Glycolysis is our fast energy system. It's not an immediate energy system, so it's not the fastest, but it is our fast energy system. In this video, we're going to be talking about how we use carbohydrate in order to make energy in the body. So let's talk about carbohydrates and what it is real quick. So carbohydrate is a molecule that combines carbon, hydrogen, and oxygen in order to make a saccharide molecule. So when we have a carbohydrate that we use in our body for energy, we can liberate about 4.1 kilocalories per gram of carbohydrate within our body. And so the most important carbohydrate within our body is glucose. So this is what we're going to be focusing on here, and that's generally what you hear about and read about in textbooks when you're reading about bioenergetic or bioenergetics and metabolism. All right, so glucose, again, the most important carbohydrate. It's a monosaccharide, which means it's a simple sugar. It only has a single saccharide molecule in it. And this is the carbohydrate that we are talking about when we say blood sugar. So blood sugar is glucose. This is also the primary energy source for the brain as well as the spinal cord. And when we do intense exercise, it is our body's primary energy source to fuel our muscles as well. So glucose is super, super important for normal physiological function, especially for exercise performance. All right, so glycolysis is the term for when we break down glucose in order to make energy from the glucose molecule. And it is going to be the primary source of energy for intense exercise from about 15 seconds out to about 2 minutes of continuous intense exercise. Another term that you should be aware of is something called gluconeogenesis. This is when we take various substances that are not glucose and we turn them into glucose. So the most commonly converted substances are going to be various amino acids, glycerol, and lactate. So all of these can be turned, as well as others, can be turned into glucose, again, through that process of gluconeogenesis, which primarily happens within the liver of the body. Our body does have the ability to store glucose, and when it does that, it stores it in a molecule called glycogen. So glycogen is nothing more than several glucose molecules that are bound together into a single large molecule. And so our primary places where we store glycogen is going to be muscle, especially skeletal muscle, and liver, within the liver's tissue. The process for creating glycogen is called glycogenesis, and the process for breaking down glycogen is called glycogenolysis. This diagram down here talks about how we can create and then also break down glycogen. So in this diagram, we have blood glucose coming past the cell, and this is depicting a skeletal muscle cell here. And the blood glucose can come into the cell. Once it's in the cell, using the enzyme hexokinase and using up an ATP molecule, we convert it to glucose 6-phosphate. Once it is turned into glucose 6-phosphate, the skeletal muscle does not have the ability to eject that or remove that glucose from the cell. So within skeletal muscle, we do not have the enzymes in order to get this back out of the cell. The liver does, skeletal muscle does not. Again, we're talking about skeletal muscle here though. So once we have the glucose 6-phosphate, it can go through this series of steps here, eventually turning into stored carbohydrate or stored glucose, again, as glycogen. Or in this case, specifically, we're talking about muscle glycogen, but it's the same as liver glycogen. The glycogen can be broken down and turned back into glucose 6-phosphate, which can then go down through the glycolysis pathway, continuing eventually making ATP. All right, so this top part right here is the same diagram we just showed when we were talking about glycogen. So this is that sort of cycle of making and breaking down glycogen right here. And here's that blood glucose coming in from the blood into the cell. All right, so whether it comes from blood glucose or comes from muscle glycogen stores, eventually it's going to turn into glucose 6-phosphate, and it's going to go down through all these steps here. This part that is in this purple box here is what is sometimes called the investment phase of glycolysis. And the reason it's called that is because we actually use energy up in order to go through these processes. Specifically right here, we use an energy molecule that was when we brought the glucose in and turned it into glucose 6-phosphate. And then we also use another ATP molecule here, going from fructose 6-phosphates to fructose 1,6-bisphosphate. And so these two steps do use up energy. This entire process of glycolysis, once it's no longer in the blood and actually in the cell, which would happen between this step here, it's going to happen in the cytosol, and it's going to happen anaerobically, so we don't need oxygen in order to do that. So that's both for the investment phase and what we're going to be talking about next, which is the harvest phase. In the harvest phase, the fructose 1,6-bisphosphate actually splits into two molecules, and when it does that, it goes down two parallel paths that are identical. Okay, so if you know one path, you know the other path. And that also means that for every molecule that is produced, so right here we're making an NAD molecule or an NADH molecule, we're actually making two of them because again, there's two pathways. So here's an NADH, here's an NADH. Right here we're making an ATP, here we're making another ATP. And then down here towards the bottom, here's an ATP, and yet again, here's another ATP. The final product in glycolysis is pyruvates in both of these parallel paths, assuming aerobic metabolism is able to keep up. All right, so looking at slow glycolysis, which is when aerobic metabolism can keep up, we end up making a net of 2 ATP, because remember, we did make 4 ATP, but in the investment phase, we used up 2, so it gives us a net of 2. We made 2 NADH molecules, and we made 2 pyruvate molecules that are eventually going to go into aerobic metabolism into the Krebs cycle. All right, so let's talk about, though, for a moment when we don't have slow glycolysis and when we actually have fast glycolysis, and what's the difference? Glycolysis can happen at various rates, so we can get what sometimes is called slow glycolysis and sometimes what is called fast glycolysis. All right, so depending on how quickly glycolysis is moving forward, we can either go slow and pump those 2 pyruvate molecules from each glucose molecule down this preferred path into the Krebs cycle, and then where it becomes acetyl-CoA, and is part of that Krebs cycle, and eventually we end up getting a lot more energy from it, which is why it's the preferred path. It goes down the aerobic pathway. If we completely fill up this pathway and we're still making a lot of pyruvate, which is what happens when glycolysis starts to run faster than aerobic metabolism, this pyruvate starts to spill over into a secondary pathway. And that secondary pathway is going to turn the pyruvate into lactate using the lactate dehydrogenase, and in the process, it actually uses up an NADH molecule, turning it back into an NAD molecule. All right, so if we do this, so if we go down this pathway here, we end up losing out on the pyruvate molecules that can go into the Krebs cycle. We also end up losing out on the NADH molecules that can go into the electron transport chain. And instead, we end up with lactate. Good thing is lactate, this pathway is reversible. So if metabolism slows down or bioenergetics slows down, glycolysis slows down, the lactate can be turned back into pyruvate, and you basically end up recreating the NADH, and the pyruvate can go down this preferred path at that point in time. But metabolism so basically exercise intensity needs to slow down enough for that to happen, or if you just stop and start resting, that will eventually happen to most of the lactate. It's important to note here that we're talking about fast and slow glycolysis here. These aren't necessarily distinct things. This is cell by cell specific. All right, so for instance, a fast switch muscle fiber, a type 2 muscle fiber, is going to have a weaker aerobic system than a slow twitch muscle fiber is going to. And so at the same rate of glycolysis, the fast twitch muscle fiber is probably going to be making lactate, but the slow twitch muscle fiber is probably going to be keeping it at the end product of glycolysis as pyruvate and sending it down the aerobic pathways. So different cells are going to produce lactate at different intensities of exercise. Let's go through a summary of glycolysis. So we start with glucose in the blood. It gets into the muscle cell or whatever other cell of the body you might be talking about, and it's going to produce 2 NADHs that are going to go on to the mitochondria to be used in aerobic metabolism. It's going to produce 2 ATP molecules that is going to be used immediately in order to keep exercise going. And it's going to produce two pyruvate molecules that are also going to go into the mitochondria in order to be used aerobically. So if we kind of zoom in on the mitochondria here, which is what we're showing down in this section, we have the pyruvate coming into the mitochondria, being turned into acetyl-CoA, goes through the Krebs cycle, produces some ATP, produces some NADHs, produces some FADH2 molecules, all of which the NADHs and the FADH2s are all going to go into the electron transport chain and produce more ATP molecules. So looking at this table here to kind of summarize what we're getting out of each part of the process. We have glycolysis that's going to produce 2 ATP molecules, 2 NADHs, and 2 pyruvate molecules. Again, assuming that aerobic metabolism is keeping up with glycolysis. We have pyruvate decarboxylation, which is a process I didn't really talk about a whole lot here, but it's actually this process of turning pyruvate into acetyl-CoA that's going to produce 2 NADHs right here, which is what's listed here under the other products of pyruvate decarboxylation. This is assuming that we're breaking down the whole glucose molecule, because remember, when we go through glycolysis, it actually splits into 2 pyruvates. So it goes through this process, then this process twice. All right, so the Krebs cycle, again, going through it twice, we're going to produce 2 ATP molecules in total from 1 glucose molecule. Also 6 NADHs, 2 FADH2s. And then once the NADHs and FADH2 molecules get to the electron transport chain, that's where they get used up, and they produce 28 more ATP. So these additional 30 ATP molecules are only going to come from glycolysis when glycolysis is slow enough for aerobic metabolism to take in the pyruvate and use it to produce additional energy. So that's why we get so much more energy out of slower glycolysis per unit of glucose than we do out of faster rates of glycolysis. Here I just did a very brief overview of the Krebs cycle and the electron transport chain. I'm going to have an entire video that I'll link to in the description below talking about how these processes work and exactly what they do. I'll also put a link in the description below to a video talking about how fat metabolism is used for exercise and how much energy and how we can get energy out of that as well.